Okay. Hello and welcome to our second seminar of week 15. For those of you that are joining us uh, from other parts of the world, we are broadcasting the seminar of the Joint School of Nanoscience and Nanoengineering via YouTube. The Joint School of Nanoscience and Nanoengineering is a unique collaborative academic unit between North Carolina A&T State University and the University of North Carolina at Greensboro that builds on the strength of the two universities to offer cross-disciplinary graduate-only programs. JSNN fosters interdisciplinary research on emerging areas of science, nanotechnology, engineering, and synthetic biology. Today, it is my honor to host Dr. Laura Miller, who is a professor of mathematics and biology at the University of North Carolina at Chapel Hill. Dr. Miller received her Bachelor of Science degree in Biological Sciences from University of Chicago, a master degree in zoology from Duke University, and she received her PhD in mathematics from the New York University. Today we'll learn more about her interesting research, which is the nature of what we have at the Joint School of Nanoscience and Nanoengineering, combining different areas of research, and more importantly, biology and mathematics. I'll remove myself from the screen and Dr. Miller will start her presentation. But meanwhile, if you have questions, please type that in the YouTube channel and I'll monitor that. And so I'm expecting that by the end of the talk, we can ask the questions that you're going to type in the chat. Dr. Miller, please.
Hi, thank you so much. So I'm happy to be here. And just as a warning, my internet connection is a little bit unstable. I'm out in the country, and you might also hear my dog in the background, but I guess these are the times that we're living in. So I'm happy that I'm able to join by video and over YouTube. And I'm going to tell you today about some of my research that is looking in particular at fluid and airflow through hairy appendages of small animals. And there are a lot of different contexts where this comes up. It's quite important to exchange and locomotion and other fundamental processes. And another interesting fluid dynamic phenomenon is that the flow can either move through these hairy appendages like a leaky rake, or if the hairs are spaced close enough together, the whole appendage can act more like a solid plate. And so I'm going to start by telling you about some important dimensionless numbers for these processes, and then tell you a little bit about the numerical methods that we use to solve these problems. And then we'll look at flows through porous layers, so how we can model something in biology that is very branched or hairy as a homogeneous porous layer. And then we'll look more specifically about how you might model the flow in between individual hairs. And then we'll talk about how these flows are important for exchange of nutrients and wastes. Now, recently, there's been a lot of interest in fluid dynamics combined with math and biology across many different scales. And the movie you're seeing right now is an old simulation of blood clotting by Aaron Fogelson. And you see platelets that are activated, they're binding together, and they're forming a clot. And so the flow will eventually stop due to the interaction between these flexible platelets and the fluid. All right, so now our clot has mostly formed. At the larger scale, you will also see fluid dynamics used in animal flight, animal swimming, at the scale of, say, feet or even meters. And an interesting thing about all these problems is that they really span a large spatial scale between the cellular and subcellular scales, all the way up through the macro scale, particularly if you're thinking about whale swimming or large bird flight. Now, in my lab, I work with students from biology, engineering, physics, mathematics, computer science, and we combine both experimental and computational fluid dynamics with math biology. So in my lab at UNC, we actually have a saltwater system for marine invertebrates where we take care of jellyfish, corals, tunicates, and other marine invertebrates. We've started doing some exciting field work in Key Largo at the Key Largo Marine Lab. We also have a number of flow tanks from the desktop scale to the scale of an entire room, where we drive flow through organisms and also 3D printed models of organisms. And then these experiments are used to motivate and validate our numerical simulations of flow. So during this talk, I'm going to tell you about both our experimental results and our modeling and simulation results, and really how they enhance each other. So let me introduce you to a couple of important dimensionless numbers in fluid flow. And the first is the Reynolds number. So if we take the Navier-Stokes equations describing viscous fluid flow and non-dimensionalize them, so we pick L, which is a characteristic length. In a biological process, this might be the diameter of a pipe, or it could be the length of a wing. And then capital U is a characteristic velocity, so it could be your background flow speed, or it could be the average flow within a pipe or a vessel. And then rho is the density of the fluid, and mu is the viscosity or stickiness of the fluid. So if we non-dimensionalize, we end up with 1 over the Reynolds number in front of this viscous term, Laplacian U. And it's basically describing the ratio of inertia to viscous forces in the flow. So high Reynolds numbers are typically going to be macroscopic inertial flows, inertial dominated. In particular, if you think about fish swimming, insect, and even bird flight, these are typically large Reynolds number problems. And they're the type of flows that you're used to if you go for a swim. Now, on the other scale, we have viscous dominated flows. So the Reynolds number is much less than 1. And these are microscale problems typically. So flowing capillaries, swimming of bacteria, for example, and inertia is negligible. Now, this has a lot of consequences for organisms, particularly how they locomote and pump fluids. So on the left-hand side, looking at external flows in biology, again, where viscosity dominates, you have things like bacteria, unicellular organisms, paramecium, and so forth. They typically swim with cilia and flagella. And then at the large scale, you have birds and whales that swim with flapping fins and tails. At the intermediate scale, where both effects are important, you end up with a lot of these creatures with these hairy appendages. So that insect with the bristled wings is thrips. I think, unfortunately, I cannot use a pointer on this screen, but look in the middle of that figure on the left, and you'll see these two critters with hairy appendages. So the insect is a thrips. They're some of the smallest flying insects, and a copepod is to the left. So we really see these hairy appendages at the intermediate scale, and less is known about how they locomote. Now, in internal flows, if you look at the graph on the right, capillaries, alveolar sacs, and so forth are going to be viscous dominated internal flows. At the large Reynolds number inertia dominated scale, you have flows through the heart, through the trachea, and so forth. And we also have interesting intermediate scales like frog hearts, some fish hearts, bronchi, and so forth. So internal flows also span this range. And one interesting thing is that different mechanisms of pumping and swimming work at different Reynolds numbers. So in this movie on the left, you're seeing a flapper that has been placed in glycerin and it doesn't get anywhere. And on the right, you see the same flapper and it swims through water as you would expect. So this is known as the scallop theorem. And basically, if you try to swim using a reciprocal motion, then you won't have any net movement. So you flap one way and you move forward a little bit, flap the other, and you move back. So if you were trying to swim in molasses, you also wouldn't get anywhere. The Reynolds number would be low enough. And so this has consequences for the organisms we see. So we don't see unicellular organisms with something like a tail, flapping tail, or flapping fins. Instead, they use flagella, which is spinning corkscrew motion instead of reciprocal. Now, this is just one more demonstration of the Reynolds number. All right, on this slide, I'm able to use my pointer, so that's interesting. So anyway, Reynolds number above 1, we're basically spinning the outside ring, and we would expect some inertia. So when you stop applying a force to the fluid will continue to move for some time. Also, if you reverse that motion, which we're now doing, things are potentially as you expect. I'll start the other movie, which is at low Reynolds number, and it will do the same thing. So on the left, at this higher Reynolds number, you spin one way, you spin back, and the flow is not the same. So it's not reversible. Now, if you look on the right, so this is a very viscous solution. So the cool thing that happens is that the flow is reversible. And we end up exactly where we started. Now there's an incredible collection of videos online for fluid dynamics, which is now hosted at MIT. So it's the National Committee of Fluid Mechanic Films, which are really inspiring and give you a lot of intuition for fluid mechanics if you're interested. So the next number that I want to introduce is the Peckley number, which gives you the ratio of advective to diffusive transport. And the image that you're seeing is of a model of marine snow. So you have this particle that falls through water, and it some of the chemicals within it diffuse outward. So if you take the advection diffusion equations and non-dimensionalize, you get the Peckley number, which is the characteristic velocity, so maybe the settling velocity of the marine snow, times a length scale, that could be the diameter, divided by the diffusivity. So the left picture where the Peckley number is 0, the velocity is 0, and this chemical is just diffusing out from the sphere. Peckley 100, we have advective transport is 100 times that of diffusive. And you can see that the chemical is diffusing out, but also being advected in this tail, as in this case, the sphere is falling. And at 10,000, we see an even smaller sort of layer of chemical coating the sphere because it's been advected away. So something interesting that happens for organisms that are where at intermediate Reynolds numbers where inertia and viscosity are balanced, and at intermediate Peckley numbers where advection and diffusion are balanced, we often find again these bristled appendages. Okay, so studying these problems mathematically is challenging. So we cannot neglect either diffusion or inertia or viscosity in the case of the fluid flow, and we can't neglect advection or diffusion in case of looking at the chemical gradient. So and analytical solutions are extremely difficult to come by, particularly for moving boundaries. So we rely on computational methods. And a lot of the results that I'm going to show you are using a couple of different software libraries put together by myself and Boyce Griffith. And our students. So the basic idea is that you want to solve this problem where you're solving the Navier-Stokes equations with an immersed elastic boundary that represents the organisms or these hairs. And you need to find a way of dealing with the interaction between the two. So the fluid can deform the elastic boundary. The elastic boundary applies a force back to the fluid that moves the fluid and so forth. I'll show you some links to these software libraries in a moment, but they both use the immersed boundary approach to fluid structure interaction from Peskin, who originally developed it in the mid-70s, and there has been a lot of work on it since. Now, there are other ways to solve these problems, but one of the nice things about the immersed boundary method is just that it has this long history, so there are software libraries available, and a lot of ways to treat different types of boundaries, including porous boundaries, boundaries with different elastic properties. You can incorporate muscle models and so forth. But the basic idea is that you discretize the Eulerian equations describing the fluid flow on a Cartesian grid that's fixed. And then you discretize the Lagrangian equations describing an elastic curvilinear mesh or boundary. And then you come up with a way of dealing with that interaction. So for the fluid flow, you're solving for the velocity, the pressure, and the body force. And for the curvilinear mesh, you're solving for its position and elastic force density. And the way that the immersed boundary method deals with these interactions is through integral transforms with delta function kernels. And again, there's quite a bit of literature out there on this. And if you're interested in these methods and the software, I'm happy to send you references and links to various codes that are available on GitHub. So one of the challenges for these types of simulations is that while you're solving the full Navier-Stokes equations, you have to resolve the fluid flow that's near the boundary, which requires pretty fine grids. And because the way that the force spreading is handled, you also have to take a pretty small time step. Now, again, there are other methods that are faster and more efficient, but oftentimes they've only been worked out in 2D, and you don't have these open source software libraries available. Although hopefully that will change and we'll have more methods available in the future. So the idea is that at each time step, you have to solve the equations of fluid motion, define on the Eulerian grid with the elastic body force that comes from this boundary, immersed boundary. And then you move the immersed boundary at the local fluid velocity, so that enforces what's called the no-slip condition, so the boundary moves with the fluid. You look at the deformations of that elastic boundary and then calculate the body force, and then you spread that force from your immersed boundary to the Eulerian grid and solve the Navier-Stokes equations again. And so in practice, you might have to do this for 100,000 time steps and on grids that are 512 squared, 512 cubed in order to get sufficiently accurate results. Okay, so again, one nice thing about this method is that people have worked out ways to deal with treating the boundaries as a series of linear, torsional, or nonlinear springs. As beams, you can add additional mass to the boundary. You can make them porous surfaces or layers. There are muscle models that have been incorporated, and you can also move the boundaries with a preferred motion. So just because of the computational cost of all of this, if you're really going to go into 3D, you need to have adaptive mesh refinement for a lot of problems and you need it to be parallelized, and you typically need to run these on clusters. So Boyce Griffith has spent probably the past 20 years, I would say, developing IBAMR, which is the immersed boundary method with adaptive mesh refinement. And so this is a swimming jellyfish. And what you can see are the different grid levels. So the finest grid is near the jellyfish and the shed vortices. So the colors you're seeing are basically showing you the vortex wake. And this updates in time as the jellyfish is swimming. And so this software library has really made these types of simulations feasible, whereas beforehand, you were a bit limited once you went to 3D. So one challenge about using IBAMR is that it requires right up to 10 third-party software libraries that you have to have the right versions, and it can be challenging to install. It's written in C++, and you have to be relatively good at programming to make changes, which isn't a problem, if you are training graduate students who are sort of computationally sophisticated and know how to program. But if you want to introduce this more at the undergraduate level to a variety of different majors, we've also written up an immersed boundary code in 2D that's available on MATLAB and Python. And so here are a couple links to papers that introduce that software library. Okay, so now let's transition to some of the applications. And as I mentioned before, there are many different types of flows through things that are like cylinders in biology. So nose hairs are one. They probably have a significant role in filtering the air that we breathe. In fact, if you remove your nose hairs, then you're more susceptible to sinus infections. But how they filter is not really well studied. And in fact, most simulations of flow through the airways ignore these nose hairs. Better studied problems include insects. So if you look at the center picture on the top, that is a parasitoid wasp. It's about 300 microns in body length. And almost all insects that are at the millimeter scale and smaller have some sort of bristled wings. So it's something that has evolved multiple times and presumably has aerodynamic benefit. And then we also see these in organisms, aquatic organisms that swim, and also those that filter feed. There are hairy structures on leaves that change the boundary layer around the leaf and affect evaporation and heat dissipation. And also internally, so the middle picture on the bottom are microvilli. Now, the flow through arrays of cylinders at low Reynolds numbers is non-intuitive. So these are basically models of just push pins. And let's see if I can pause this. So what is happening is that the fluid is corn syrup. The flow is going to move from left to right. These are push pins. So there are rows of them. They're spaced apart along the row, about the same spacing in between each row. So they aren't packed together. And then these are lines of dye that have been drawn in the corn syrup. And as you turn on the flow, you can see that those pins almost stop the flow through the rows. Now, if you have a higher Reynolds number flow and pins that are spaced further out, then you get more flow through that layer. But the amount of flow within the layer is going to depend on the Reynolds number and the spacing between those pins. And this is just a neat example from an undergrad where you can see that there's some interesting parabolic flow in this less dense layer, since on top, gems have been used to make this a denser layer. So interesting spatial effects that can happen from changes in density. Now, let's look at some biological examples. And so, again, like I mentioned, there are different ways that we can treat these problems. So we can treat the porous layers as, or the sort of bristled layers as a homogeneous porous layer, or we can try to solve the flow between each. One example of treating this as a porous layer, which is the case of upside down jellyfish. So on your left, upside down jellyfish basically live on the bottom of shallow parts of the ocean. So they're in the Keys, and they sit with their bells upside down on the substrate. And then when they pulse, they form these beautiful vortices that you can see on the left that basically they're using this, creating these currents for filter feeding. And so if you're plankton, a copepod, you might get wrapped up in that vortex and then push through these oral arms. And we'll look at a better picture of those, but that's kind of the grassy stuff you see on top of the jellyfish. And so that's where they will sting, capture them in a mucus net, and then eat them. All right, and also on the right are brine shrimp, which are something that the jellyfish eat, but their appendages are also bristled, which you may be able to see in this video, but might not have enough resolution. So again, we see these structures all over the place. And so here's a top view of upside down jellyfish, and it is the oral arms, which you see from above, are incredibly complicated. So this jellyfish, the mesh that you see, each spacing is about 1 millimeter. And so this jellyfish is probably 6 centimeters across. And then as we zoom in farther and farther to these branched oral arms, you see that there are even smaller scale branches. And if you look at the panel on the right, each of those kind of flowery structures are probably around the 500 micron scale. So it's incredibly multi-scale problem with a lot of morphological complexity. And there's probably extremely interesting fluid mechanics that's happening in between all of these branches, which is going to be difficult to resolve. And our group has yet to have done that. It's also difficult just to visualize the flow because the branches are opaque. But we can do some things to look at more of the bulk character of the flow and treat this oral arm structure as a porous layer. Now, this is a video using particle image velocimetry of an upside down jellyfish. So the arrows show you the direction and magnitude of flow. And you can really see that starting vortex that forms during contraction that's going to wrap up plankton into the vortex that then pushes it through the oral arms. And another feature of the flows that they set up is if you look above the jellyfish, there's kind of a constant slow upward jet. So the jellyfish is essentially pulling in fluid from its sides along the substrate and then pushing it up and away through the oral arms. And so it's constantly sampling new fluid. So this is a high Reynolds number problem. If you put a jellyfish in low Reynolds numbers, this is not going to work. So you would pull fluid in during this contraction, and then you would push it back away during the expansion. And we were able to get some quantitative results using PIV just for what this flow structure looks like. So again, you can see this flow has been averaged over the whole cycle. So you're pulling fluid in radially and then pushing it upwards. And the lines on the picture on the left correspond to the lines in the graph on the right. So there is a sustained upward jet as you move up and away from the jellyfish. And that peak flow actually corresponds to where all the mouths of the jellyfish are located. So the whole structure is really designed to pull in new fluid, push it up through the areas where all the mouths are. And they have about 50 or so secondary mouths. So my first graduate student, when we were starting on this project, we wanted to come up with a simple model to try to describe this pattern. And again, we weren't ready to try to simulate flow through those complicated oral arms. So we just treated the oral arms basically as a porous line that hovers above the jellyfish. So if you look towards the bottom of the screen, that's the model. So the jellyfish in pink, the edges are pieces of an ellipse that change their aspect ratio over time to generate the pulsing. And they pull in fluid and then push it through. You know, our beautiful oral arms, we basically just made a porous line. So this, you know, is a nice example of the mathematician's spherical cow. So how we dealt with the porous line, it's a modification of Darcy's law. So the force per unit area normal to the surface is going to be equal to the pressure jump across the surface. The pressure jump is proportional to the force acting on that layer in the normal direction. And so essentially, rather than having the porous layer move at the local fluid velocity, we allow a slip. And the slip between the fluid and the porous layer is proportional to the porosity multiplied by that normal force. So we can make it more porous and less porous, and the bigger that pressure jump across the layer, the more flow. So here are some of her simulations. They're just 2D. So these results were probably from about 10 years ago. So it was before we had and could do 3D. But on the left, you see a porous layer, and on the right, you see a solid layer. The little pink dots are just passive tracers in the fluid, and the colors are showing you vorticity. And so if you have a porous layer, you're basically bringing in those particles from the side, and then they circulate around the porous oral arms. And if the layer is not porous enough, then the fluid just jets out from the side. And another interesting feature we found from the simple model, so this is showing you the upward or the flow towards the jellyfish, where that flow line is over time. The peaks correspond to each contraction. The pink line is the biologically relevant case where you've got porous oral arms at about the right Reynolds number of 450. And the green and kind of purple dashed lines are at Reynolds number 45, so it's lower, and it's actually lower than where you would find these jellyfish. So you don't find them that small using this type of behavior. But only if they're big enough and actually have porous oral arms do you, by the end of this fourth pulse, start to see continual flow, almost continual flow towards the jellyfish, even when the bell is at rest. So the peaks, again, correspond to the contraction. The depths correspond to the expansion, and then there's a rest period before the next expansion. So the presence of these oral arms in a sufficiently large size is needed to set up sort of this continual flow towards the jellyfish. And we were able to get this again from an extremely simple model that was informative. Now, other types of porous layer are protective layers that again come up in many different contexts. And you can think of beds of seagrass and corals as providing sheltering environments for plankton. So out in the ocean, you'll have wave action back and forth, and very tiny organisms on the millimeter scale that really want to stay within the coral reef or the seagrass bed where there are nutrients and other plankton rather than getting swept out into the open ocean. So these structures can really make a sheltering area. And again, if you look at the bottom right, these trichomes can really change the boundary layer along leaves, which might be useful for water conservation, heat dissipation, and so forth. Trichomes are also used for defense, so they do have a lot of other purposes, but at least in some cases, they do seem to alter the boundary layer along the leaf. And again, these protective layers come up at all scales, internal and external. It's thought that the glycocalyx, or the endothelial surface layer that lines the inside of all of your blood vessels, shields the endothelial cells from shear and creates a microenvironment external to the endothelial cell. And that's at a very low Reynolds number, about 10 to the negative 3. Again, microvilli at the intermediate scale are bristled wings, trichomes. Acetasts, I'll tell you about later. They're used for snipping. And then at the larger scale, again, we have sea grasses, sea oats, coral reef beds that can serve as these sheltering layers. And actually at the micro scale and at the larger scale, people have used the Brinkman equation to model these protective layers, as well as other approximations. But basically what you do is to take the Navier-Stokes equations, and then you add this term u alpha squared u, which is sort of a fudge factor. So alpha is the inverse of the hydraulic permeability. So if it's 0, then there's no porous layer present. If it's positive, there's a porous layer. And this term essentially applies a force to the fluid that slows it down. And you can create regions, where alpha is varying in space. And so you could have a region that's a Brinkman porous layer, and then sort of no porous region of the flow as well. And you can also come up with a poroelastic immersed boundary method, where basically you make the porous region out of boundary points that are connected with springs. And then you assume that the elastic force and the Brinkman force are balanced, and come up with a slip, again, between the velocity of your porous layer and the background flow velocity. So this is going to allow some slip between the two. And so we've looked at this a bit at very small macrophyte or macroscopic plant beds and streams and lakes. And we measured the average flow by resolving it between basically each cylinder, and then compared that to the Brinkman model, and had very good agreement. So that's nice to see. Brinkman model has also been used at the small scale, so looking at flow within vessels and capillaries and how it's altered from that endothelial surface layer. I think there's some sound on this, so my apologies, but I don't know if I can mute it. Anyway, hope it's not too bad. So this is another experiment where we're looking at how these cylinders might create a protective layer for plankton. And what you're seeing injected into that flow are brine shrimp. So we've been counting the distribution of brine shrimp in a background flow with cylinders at different densities, and comparing that to mathematical models. And then we've been developing agent-based simulations to show plankton and other brine shrimp in flow. And this is really a work in progress, but I did want to at least show you the preliminary results. So you can see that the little blue dots on the left are plankton. They get swept away if they're outside the layer. whereas some of the guys who are in between these cylinders that could be representing blades of seagrass or something, they're able to stay for longer. Okay, so let's transition and look at actually resolving the flow through each cylinder, and looking particularly at tiny insect wings. So again, these are all very tiny insects that have bristled wings. They could be as small as 300 microns in body length. So fairy flies, thrips, parasitoid wasps are included in this category. Here's one of them in flight. They flap at 200 frames a second, and this was filmed at 4,000 frames a second. So one of the things to note from the last video, again, is that most of these super tiny insects have these bristled wings, and they clap them together and fling them apart. So one thing that's been known about such insect wings and other critters that have such bristled appendages is that they often operate in this region where they can quickly transition to a lot of flow going through the bristles versus not much flow. And that depends on the velocity that they're moving and if they interact with other boundaries. And Shear and Cole introduced this metric called the leakiness, which gives you the ratio between the average velocity of flow in between the cylinder, which you can see on the right, versus how much flow would move through if the flow was inviscid or didn't have any viscosity. And so a leakiness of 1 is totally leaky, like those cylinders weren't present. A leakiness of 0 would mean it totally stops the flow. And if you look at the graph on the left, that really depends on the spacing. So this assumes the cylinder diameter is 1. So as you get up to spacing of 50 to 1 gap to diameter, you'll get more flow. And it depends on the Reynolds number. So the higher the Reynolds number, the more flow. But you can see around spacing of 15, let's say, that if you change the Reynolds number, which could happen from moving the wing faster, you jump from a leakiness of 0.4 to almost 1. You could also change the spacing by letting the bristles flare out during part of the stroke and contract during another, and that happens as well. So for this problem, we've also combined experiments and numerical simulations, and we have a flapping robotic wing where we can measure the flow between these bristles, and this is work with Arvind Santana Krishnan at Oklahoma State. Here are some of his movies. And so these are wings that have been immersed in bliss. And you can again use particle image velocimetry to obtain spatial and temporal resolved flow fields between them. I'm going to tell you what our results look like for 2D models. And again, it's very similar to Arvind's result with his 3D model, physical model. So let's assume that you have an insect and you take a slice along the tip of the wing. So you could treat that as a row of cylinders that is flapping back and forth in space. So this is our first approximation of how to deal with this. Again, if you're in 3D and you're trying to solve the flow between dozens or even hundreds of bristles, it's a pretty computationally challenging problem that we're trying to work towards. But for right now, 2D, you're able to get really nice resolution between these bristles. And the thing that you'll notice, as you might guess, is that as you increase the spacing, so if you look on the right, there's a spacing of 5, 11, and 17. On the left, it's basically a normalized lift on the top and normalized drag on the bottom. So as you increase the spacing, the forces drop, which makes sense. You have more flow between these bristles, and so the force is going to be reduced. And so this had been found before. The question was sort of that people asked was, well, if you have these bristled wings, the only thing they're good for is just reducing both lift and drag, but you're trying to generate lift and drag. So, what is the purpose here? But so I'll point out that from that first video, again, a lot of these insects clap their wings together and fling them apart. So wing interaction becomes important. And at the top, to model this, we have solid plates. So 2 solid plates on the top left and 1 solid plate on the top right. They're moving with the same motion, so away from each other, or just one plate moving to the right. And on the bottom, you might be able to see these little dots. And that's your array of cylinders that's representing this porous wing. And so the colors, the red corresponds to the velocity of the wing, pink is faster than the velocity of the wing, and then blue and green is lower. So on the bottom, the wing is moving through the fluid and not generating as much flow. And this effect is stronger with 2 wings than it is with 1. So that you can see that by comparing the bottom left and the bottom right. All right, but what does this mean? So on the left, this is a log scale of the force. So differences that you see are actually pretty major differences. And the solid lines are 1 wing and the dashed lines are 2 wings. Again, they're moving away from each other, just translating. And there's this huge peak if you have 2 wings. And okay, so the colors, maroon is a solid wing. So the maroon, 2 solid wings is the maroon dashed line, and it's always the biggest peak. And again, it's huge because this is a log scale. But as you make this not a solid wing, that peak force really goes down. And so if you watch these insects in flight, particularly the ones smaller than what I showed you, even with the bristled wings, they clap the wings together and it's almost like the wings get stuck for a bit before they can fling them apart. So they're flying at such a small scale that air is effectively so sticky that if you take these wings and stick them together, it's going to be really difficult to then fling them apart. But if they're bristled, some flow can move between the bristles and they are actually able to fling them apart. And then once the wings are completing the rest of their stroke, they do act mostly solid, so they are able to generate force. So that seems to be the trick. Again, this isn't a problem for like fruit flies or butterflies, but for these super tiny guys who are 500 microns, 300 microns, it's a really big problem. And it seems like they're solving it again with the bristled wings. Okay, but again, at other scales too, we see these bristles. And on the left are coral polyps that actively pulse. And on the right is a crab that's sniffing. Okay, so some fun music with that one. So both of these, on the right, the crab is trying to uptake chemicals to sniff. And on the left, the corals are trying to get rid of excess oxygen for the symbionts that photosynthesize and live within their tissues. So let me tell you first about the crab sniffing. And so basically crabs, we've got a hermit crab in air on the top left and a blue crab in water on the bottom left. And they have these acetass arrays, which are basically arrays of cylinders or plates. And they flick these acetass out in order to sniff. And Lindsay Waldrop on the bottom right looked at these guys for her PhD thesis and then came to UNC to work with me, and also Shilpa Khatri on the top right joined us. And we wanted to take what she had done experimentally and do some simulations. So there's a big challenge with this problem, though. The blue crab acetates have about 400 bristles. Now, Lindsay, for her PhD work, sort of amazingly made a physical model of these acetates, which you'll see on the right. She learned how to pull glass and make a similar model. She also made the glass match the refractive index of glycerin. And one thing I think I forgot to mention is that if you want to look at the flow through these experimentally, it's great if you can make it larger scale, but you want to keep the Reynolds number the same. So you've increased the length scale, you compensate by increasing the viscosity. So rather than using water, you might use glycerin or corn syrup. All right. And then she's made them transparent so that she can actually visualize the flow through them. So what they end up doing is during the downstroke, the hairs are typically a little bit flayed out. And you can see that on the leftmost panel of the model. And then during the return stroke, those bristles collapse, which you can see in the middle panel. So they're changing the leakiness in this case by changing the spacing between the cylinders. And she used the physical model and PIV to measure the flow in between those bristles for the different models. And she did that for blue crab models and hermit crab models at Reynolds numbers set for air and water. So this requires a little bit of explanation. We wanted to understand how this was helping with chemical uptake. And so we wanted to use an advection diffusion model, but we really couldn't simulate the flow through 400 bristles at an intermediate Reynolds number where you really have to resolve the flow. So we used her Lindsay's PIV experimental data as the background flow field, as the input velocity for our advection diffusion model, and then used chemical concentration either as a filament, or as a region. And so if I play this, what you'll see, the bristles are played out and the concentration moves through these tiny white dots. So it's uptaking this chemical. Now you'll see a reversal. So they're trapped. All right, so we changed the leakiness. So it was leaky enough the chemical can go into this array. And then during the upstroke, it's trapped and it allows time for those acetates to uptake the chemical. And it's similar if we just have a whole region rather than a filament. So the array is really well tuned for doing this. Does this pattern help the uptake? And so what we found, the uptake over time, blue is water, red is air. And so if we just look at the blue, this is the blue crab morphology in water. And it does a much better job of uptaking our chemical than the hermit crab, who's typically in air. Now, if we compare that to what's happening in air, the hermit crab that lives in air does better than the blue crab that lives in water. Air is always easier to sniff in than water because of the diffusivity of the chemicals that they're sniffing. But each organism is tuned to its particular environment. And again, we see this pattern where we want to bring in new fluid to sniff and then hold it there for a while to sample. And I don't have much time left, but I'll just tell you that some of the highlights of the soft pulsing corals, which is a similar story. So they pulse their tentacles to enhance exchange. They have these symbiotic algae that live within their tissues that photosynthesize. And when they pulse, it actually increases photosynthesis by up to an order of magnitude. And this seems to be because it allows them to remove O2, which is a waste product of photosynthesis, so that that reaction can increase. So here they are using PIV. So you can see that they're generating these jets. So again, they pull in fluid from the sides and then push it upwards, similar to the jellyfish. And the fluid moves through these bristled tentacles. We used, in this case, 3D simulations to model what was happening. So a contraction and an expansion. And again, the fluid is being pulled in between the tentacles and then eventually up and away from the tentacles. Oops. These guys operate at Reynolds numbers around 10. And it ends up that if you change the Reynolds number, they're actually tuned into a peak of the max volumetric flux upwards and max horizontal flow. So if the Reynolds number is much lower, we start getting into these reversal problems where you pull in fluid and then push it back and you never bring in new fluid. And if it's too high, then the region of fluid brought in becomes smaller due to the decreased viscosity. So these guys really do seem to be tuned to their size, which again is probably about a half centimeter in diameter and a Reynolds number of about 10. Now you can also consider exchange that's happening on these boundaries. And so Shilpa Khatri and her student Matea at UC Merced developed an immersed boundary style approach to looking at the advection diffusion equations where our immersed moving boundary is either a source or a sink of a concentration gradient. And I'll just quickly show you some of the initial results. So they're developing and testing this method. Right now it's in 2D. We're changing the Peckley number. So you can see at higher Peckley numbers, we do have some advection of our concentration up and away from the coral. At lower Peckley numbers, this is really diffusion dominated. And so this approach of actively pulsing would not be as effective. The corals are probably around a Peckley of about 100 or more. And so this is the scenario that we're looking at. So you could think of these tentacles as generating oxygen as a waste. And then the tentacles are basically infecting that oxygen away. And then we have a lot of future work planned looking at different types of corals, ones that don't actively pulse, but instead have flow that moves through them, such as these sea fans that you see here. But this is, again, some work in progress. So I'll just end there since I'm pretty much out of time. But there are many interesting and different types of questions that come up looking at porous layers in cylinder regions in biology, both for locomotion and exchange and sheltering. Some of the really interesting questions are when you balance inertia and viscosity, and also when you balance advection and diffusion. And there are a lot of very challenging, both computational and mathematical modeling questions. And then just to end on the acknowledgement slide, so many different people have helped with this, again, from all disciplines and from undergraduates to faculty, and all in there. So thank you.
All right. Thank you very much, Dr. Miller. Fortunately, we didn't get any technical difficulties. I was monitoring the YouTube channel, and it seems like we didn't have a problem. Now, I know that there is a journal club that some of the students and faculty want to participate, but we try to be on time. So anyways, I do have one question from one of the audience, and that is there a porosity scale at which the assumption of the homogeneity no longer holds?
Yeah, I mean, I think it really depends on sort of the context that you're looking at the problem. So for all of these models, if you really need to resolve what's happening in between the cylinders, like if you're looking at chemical uptake and the boundary layer around the cylinder really matters, it's going to be difficult to get at that by just homogenizing the layer in that way. I also think that, if your array of cylinders is really, the spacing is non-homogeneous so that you have some gaps, much larger gaps, or they might not even have to be that much larger if you're at this leaky to solid transition. It's not going to be a good approximation because you can imagine, slow flow in certain regions and sort of quick flow that's winding in between these larger gaps. Possibly you could handle that with, a spatially varying porosity, but it might be challenging. And, I haven't looked at that carefully.
It's indeed interesting. The second question that I have from the audience is, to simulate a porous layer, is it necessary that the layer be periodic or continuous, or can it be random?
Yeah, so it does. I think it could be random. Again, at some point, it may no longer serve to be a good model, right? But you could definitely look at highly spatially varying porous layers that are irregular, or appear in different regions rather than just like a block, let's say. You probably, depending on how irregular it is, I think you would want to go in and check whether or not you were describing the flow patterns reasonably or not.
Yeah. So another question from audience is again, I guess this one is on so they ask, on smaller scales, how the surface tension forces are taken into account in numerical simulations.
Yeah, so at the scale where surface tension becomes important, I haven't worked too much in that. So with the insects that I've looked at, it's relatively a small effect, although if you get much smaller, it could potentially be important. I know that there are ways of handling it. I haven't myself done that, but there is, I think Shilpa Khatri has worked on, if I remember right, like an immersed boundary style way of handling surface tension. But yeah, I know that people have done it, but I don't know the details.
Okay, that's fair enough. Well, I have a question regarding, I think that was the last question from the audience, but I have a question regarding the models that you're using. And so generally, do you think that these models could be modified to work with particles in the air, and that is aligned with distribution of the nanoparticles, for example, in the environment?
I think so. So I think, depending on what particles you were looking at, you would need to potentially consider electrostatic effects as well. And there are some people who have been working on incorporating electrostatic effects into the immersed boundary method. So, I mean, it's not too hard to do it just in a 2D simulation, but in 3D it gets tougher just because of what the computational cost is. And so you would need some way of, deciding whether or not you would calculate electrostatic attraction or repulsion for different boundary points depending on if they got insufficiently close. So you would need to tag those points. Again, it's not impossible, but it takes a little bit of work to do it efficiently.
Yeah. So in terms of the algorithms that you're using, is it easy to modify them accordingly, or is it so the question that I have, and it is a continuation of the last question, is would you need to change parameters, and that's the difficult part, or is it the changes that you need to do to the algorithm that is more difficult?
Yeah, I mean, I think, a lot of this depends on context, like what scale are you looking at? What are the density differences between the particles and air? So, the algorithm, there are methods for, again, like looking at additional mass of a particle in air, let's say, which isn't a problem if the density differences are small, but then there becomes a stability issue when those density differences become large, so that it no longer becomes computationally tractable. And so then you have to come up with a new numerical method, which, you might be able to find some that already exist, but then you'd have to implement them into IBAMR.
Got it.
Guess, does that answer the question?
Yeah, absolutely. And thank you very much. Thank you for doing this seminar. It was really interesting, and it shows how little we know about biology, right? I mean, this is one of the things that sometimes we think we know a lot, but I mean, your research shows that we basically don't know much. It's just at this point we need to do more to understand biology. But anyways, I really appreciate you coming up and doing this seminar for us. Hopefully, we're gonna have another seminar in future that will invite you, and you'll be kind enough to accept the offer. But thank you
Yeah,
very much.
definitely.
So we're gonna
Yeah, thank you.
we're gonna close the broadcasting right now. Please stay online. I'll talk to you.
Okay. So